Hi, yes, I'm Catherine Pettinger. I am from the University of Wisconsin-Madison. I'm a graduate student in the Erica Majumder Lab. And let me just hide that. And today I'll be talking about some microorganisms at a uranium contaminated site in Riverton, Wyoming. So Riverton, Wyoming had, there we go. So a little bit of background about the site. So Riverton, Wyoming was a uranium mill operating site, and that operated in the 1950s and 1960s. What they did with the tailings is they basically put them in one large pile, and that sat there for about 20 years. And during that time, uranium, radium, and a couple other contaminants leached from the pile into the groundwater. The pile was removed, however, uranium levels still continue to be elevated. We also, and there was a lot of modeling done at the time, and it was expected that the uranium would flush naturally from the aquifer. However, that is not occurring at the expected rate. So we wanted to find out what is going on, what is influencing the rate of uranium flushing in this aquifer. As you can see from the top right image here, uranium is mainly in the plume, is mainly in the shallow aquifer. So that is a fairly oxic aquifer compared to some of the deeper layers, which makes the site a little bit interesting. To start answering our questions of like the rate of flushing, we sampled from across the site, including upgradient within the former tailing areas, off-gradient, and downgradient, and did a series of microbe tests along the redox gradient. So some of those results, just to kind of look at what, and get a conceptual model of what can happen to uranium. So uranium exists in oxic water, usually as uranium-6, which is in the center here. It's fairly mobile, aqueous phase. However, it can be reduced by microbes in deep subsurface. So we wanted to test, first of all, and that would change it to be more of the sedimentary phase. Does that reaction occur? And we saw from the microbes in our lab tests that that wasn't quite occurring at in laboratory conditions, at least. So then uranium can also absorb or stick to lots of different coated minerals. So we went through, and although those are usually by abiotic processes, the formation and dissolution of the coatings are often contributed to by biotic processes. So for example, starting with we're going to kind of work left to right. Manganese, we did see some reduction and oxidation of manganese, suggesting that microbes can contribute to the formation and dissolution of the manganese coatings, which uranium could potentially absorb to. And then sulfate, we saw sulfate reduction as well as sulfide oxidation. And there is lots of articles about iron sulfides, also frequently called pyrites, and uranium absorbing to these pyrites or them acting with uranium in various other fashions. And then looking at our, kind of moving a little bit to the right of that, uranium oxidation and reduction. So this one was a little bit interesting because we saw oxidation in a couple sites, but not frequently, but usually the ones that had like surface water inputs. Which makes sense that more oxic conditions, there might be some microbes that can utilize the oxygen in a microaerophilic environment. And then we did see some iron reduction in most sites. Finally, I have on the left a little bit about the nitrogen cycle. We saw nitrate reduction, but not too much ammonia oxidation. I didn't include that in the large diagram, largely because nitrate is not a contaminant at this site. Most sites with uranium also have nitrate pollution, but this one does not. So what does this tell us? Well, Riverton microbes are capable of many different reactions that could influence uranium flushing and uranium absorption. So what do we actually do with this information? So we need to find out, first of all, which of these reactions are occurring, especially during our simulated remediation efforts. There's been different remediation flushing attempts. So kind of finding out what of these reactions occur and how does that contribute to the uranium flushing, with the ultimate goal of enhancing flushing of the uranium out of the aquifer. And here are some acknowledgements, and that is all I have.